So, good morning all of you. I welcome you on today's session. The speaker of this particular session will be Dr. Saugata Hajra. He's associate professor from IIT Roorkee. In his educational background, he has done his M.Tech from IIT Kharagpur, followed by PhD in structural biology and drug designing from the University of Illinois, Chicago, USA, followed by first postdoctoral research in the Albert Einstein College of Medicine's New York, USA. Then in second postdoctoral research, he was in Jude Children's Research Hospital, Memphis, USA. Then he returned back to India, joined IIT Roorkee as an assistant professor. His research interest based on the antimicrobial drug resistant targeting beta-lactamase enzyme. He is having more than 90 publications and several patents in his credits. And he is also the course coordinator in NPTEL for the structural biology. With that, I believe you will convince in having huge experience of Professor Hajra in this particular field. So we will be talking about different aspects of protein crystallography on its role towards the drug discovery. So without any further delay, I welcome Professor Hajra to deliver the lecture and to attend the question answering and other things. So please, it's all yours.
Thank you, Sandipan, for the nice introduction. Welcome everyone. So as Sandipan told, something in adding to that, today I will talk about structural biology, in addition computational biology, and definitely their role in drug discovery. So to start with, I will try to talk about how protein folding, the fold is very important. To introduce structural biology, we want to know that why we will study structural biology. So we believe that, and a lot of people believe that, seeing is believing. I'll start with a photo. Looking at that, every Indian feel proud and be happy. So that's what the effect of something we look at. As Einstein have talked about, a picture is worth 1,000 words. So what structural biology actually shows us? Structural biology is a study of how the biological molecules are built. Using a variety of imaging and spectroscopic technique, scientists view molecules in 3 dimensions to see different aspects, like how are the architecture. If you look at this protein, this is famously known as green fluorescence protein, and how the fluorescence is restored, how the beta barrel structure is not allowing the water molecule and allowing the signal to pass through and developing the fluorescence color. And biology have changed through the innovation of the protein, green fluorescence protein. How macromolecules interact. If you look at these pictures carefully, you will see that there are a lot of protein DNA binding. Protein DNA bindings are the basics of a lot of phenomena in signal, like in response to particular signals, there are particular type of protein DNA interaction. The proteins, they are working as transcription factors. How the membranes are built up. Membranes are very complex creatures. When we talk about proteins, proteins are actually of 2 types. Cytosolic protein, which are soluble, and membrane protein. So how the membrane proteins are ingrained inside the membrane, how the membranes are allowing the solute to go inside, make them specific interactions, all of them we could understand if we look at the structure of the membrane. How the protein synthesis machinery, we all know about ribosome and the protein synthesis machinery. This is a huge structure, and here there is combination of protein and nucleic acid. They are helping the proteins to synthesize particularly and then release in a point where they start folding. Also, how protein interact with the solvent. If you see here, it's a setup where the protein is interacting with the water, with the solutes which are salts. The green balls are representing chloride, whereas the blue balls are representing sodium ions. How protein adopt alternative conformations. So, you know, protein work with a switch. When protein have to work, when protein not have to work, that determinant is the conformation. So how the protein change their conformation, open conformation, closed conformations, they are largely connected to the functional response of the protein. All those information help researchers to understand how the thousands of different molecules in each of our cells work together to keep us healthy. The structural studies have also shown how mishapen molecule makes us sick, and as a result, these studies have prompted new treatments for many diseases. So, from sequence to structure to function, it is very important to get the structural information because we want to know about the function, and from sequence it is our connectivity which comes through structure. The eye does not see, the brain sees, the eye just transmits. So what we see is not only determined by what comes through the eyes. What we see is affected by our memories, our feelings, and by what we have seen before. So I talked about seeing, but there is something more than seeing. We are now going there. So before going there, because this course is about drug discovery, I start with a simple question. What do you guys mean by drug discovery? And you know, I will like if you guys respond, only then you know we could be interactive. It's not a class, you all came, and thank you for that. So let's you know continue the interactions, try to answer. If you are involved, it will help me to go into more deep. Okay, so anyone, what do you mean by drug discovery? You don't have to stand, sit down.
Sir, drug discovery itself is sufficient to define. It's a designing of a molecule that has potency to cure any kind of disease by targeting with protein or enzyme, whatever mechanismic pathway they are following. So nowadays, minimizing the time and minimizing the toxicity effect is the main point of attraction for discussing and approaching this drug discovery process.
Anyone else?
Invention of new compound.
Your name, please?
Megha Ambatkar.
Megha.
Invention of new compound to treat the diseases or for use for diagnosis by various techniques. But in traditional way, it takes time, that is 10 to 15 years. But new approach, by using new approach, this time is reducing.
Okay, so you know the chemist in us makes the definition little bit in a different way what you guys are told. We say creating small change in chemistry and which leads to or which greatly affecting biology. To support what I am trying to say, I will talk about a case study where a small change in chemistry will affect biology. So all of you, I hope, knew that structure. This is DNA, which is made of the bases, sugar and the phosphate groups. Now, if you look at, there are different nitrogen bases: adenine, guanine, thiamine and cytosine. So, to develop a double-stranded DNA, adenine form 2 hydrogen bonds with thiamine and cytosine form 3 hydrogen bond with guanine. Now, think about how much our life is dependent on them, right? So, what if the situation change a little bit? So, look at there. Here, I am picking up that G triple bond C, guanosine triple bond to cytosine. But in case of this cytosine, which is actually deoxycytosine, I am introducing a molecule which is dideoxycytidine. What is the difference between them? Between deoxycytidine and dideoxycytidine is the hydroxyl group, right? So, now if only hydrogen bonding is the base, then deoxycytidine make a hydrogen bond, dideoxycytidine will make exactly same hydrogen bond, right? Everyone, you could come in front. Why? Come, please. नहीं, अब मैं सबको बोल रहा हूं, प्लीज 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 कम. इतना जगह है किसके लिए? So now you understand that in case of deoxycytidine, which is a physiological molecule, if there would be a non-physiological molecule, dideoxycytidine, it would be making same hydrogen bonds. But what would be the effect? So this, if this is a normal chain elongation process of DNA synthesis, here when this dideoxycytidine would be introduced, there is no further chain elongation. You all agree to that? पीछे कोई response नहीं देख रहा हूं मैं. So when it comes to, you see that our elongation would be stopped, right? Which would be further leading to chain termination, further leading to cell death. So basically, when I say this condition, you now probably might think that why this is not happening in the body, right? Why we are not frequently having such molecules coming from food or something and go to the DNA and making DNA terminated like that, right? So body have a very interesting process for that. What is the interesting process? You know, whenever a reaction happens, the del G have to be negative for a spontaneous reaction, right? If it is not a spontaneous reaction, what you do? You provide heat or any other external stimulus, right everyone? But when it is body, what should we do? We take food and then how to like add heat or something extra. So there are many biological reactions which are del G positive, especially the polymerization like DNA elongation. these are always a reaction which are thermodynamically unfavorable. What nature do to make them favorable? Nature spends some energy currency in terms of nucleoside triphosphate, ATP. So they couple the reactions so that with the negativity coming from the ATP, the overall reaction which is del G positive now become negative and the reaction goes on. Okay, that's the general way. Reaction A plus ATP to AMP conversion and the overall reaction is favorable. Here nature don't do that. It takes even a smarter way. What is the smarter way? They make the deoxycytidine to deoxycytidine triphosphate. Why? Then they make sure that cytidine triphosphate is not coming as a triphosphate in the cell. You understand what I mean? So because the cell membrane is, what is the nature of cell membrane at the outside? Negative in charge, right? Everyone? You have idea about that? So outside the cell it is negatively charged. So a molecule with triphosphate cannot get entered there. So it could only take entry through the form of deoxycytidine. And it have to be triphosphorylated specifically inside the cell. By that, two things happen. One, they don't get the triphosphate of any form and they could do a selection while phosphorylation. In that way, they make the selection in a positive direction. But the interesting thing is now, they make this triphosphate. This triphosphate, how it is utilized? It goes to the running chain in the form of a monophosphate it added and 2 inorganic phosphate release with the release of the energy. Everyone clear? So, dideoxycytidine cannot get enter if not the things are given in a different way, and the different way is therapeutics. How? So, as I told, it comes from this triphosphate. Now, this nucleoside analog, because the structure of this is like a nucleoside, but it is not exactly a nucleoside, so they are called nucleoside analogues. Nucleoside analogues are compounds with identical hydrogen bonding properties of natural nucleosides. When incorporated, they use this property to block the synthesis of DNA chain leading to cell death. You all agree to that? Here, the nucleoside analog is working. Like, we are not allowing the natural ones to get, but we could use that as a drug when we want. We incorporate that in a special way. Okay? This is a classic example of small change in chemistry. What is the change? Just the absence of a hydroxyl group, right? And that lead to cell death. So that's we call small change in chemistry, dramatically changing biology. So it happens, but idea of other drug designing also coming from here. So this is a nucleoside analog. Why we are using it? Because we have a target enzyme. What is that enzyme? This enzyme is deoxycytidine kinase. Anybody hear about deoxycytidine kinase? Anybody seen people getting chemotherapy? So when people are given cancer chemotherapy, major target is this enzyme, this deoxycytidine kinase. Why? Remember I talk about this 3 phosphorylation. Deoxycytidine have to be phosphorylated 3 times. It is with monophosphate kinase, diphosphate kinase and triphosphate kinase. The first enzyme is the most important, and that's why it is used in cancer therapy. But in case of this enzyme, deoxycytidine kinase, the more interesting thing is it's a broad spectrum enzyme. Now you have idea about broad spectrum, narrow spectrum. So enzyme, what we are going to talk about, what we are going to look about, have a 3-dimensional structure. In that 3-dimensional structure, in the core, there is a active site. That active site might be specific to one compound or might be broad specific, like to different type of compounds, or might be non-specific. So it could behave in a very different way depending on the active site, volume and the chemistry inside. So, if you look at this year's chemistry Nobel Prize, one of the guy was David Baker. Anybody hear about protein engineering, David Baker? No? So when I joined my PhD in 2004, I had opportunity, that time I'm coming here, there was a project which called Human Genome Project. The way genome started readout, and US get the entire credit of that. I'll show you through few slides. When those things was happening, they thought, why not going ahead? Now we could know each and every properties of the DNA which convert to the property of the protein, right? Now, is it possible to change the proteins, the enzymes, and use them in different way? 2 people came out. One, Francis Arnold. What Francis Arnold did, what in biology, a lot of you who are doing biology, thinking about, reading about, taking a protein, looking at its function and changing the function by changing the amino acids in a way called random mutagenesis or site-directed mutagenesis. People, if you are not acquired, please raise your hand and ask, sir, define me this. So you all know what is random mutagenesis, what is site-directed mutagenesis? No? Please pick up. Otherwise, you will come, I will say, I will go. There will be no meaning of that. Please pick up, please. So, the protein coded from a DNA. All the changes you want to do in a protein, you have to go back and you have to make in DNA. When you know about the protein structure, when you know about the protein's detail, and by knowing you are changing a particular amino acid by changing a nucleotide, that is called site-directed mutagenesis. Whereas you have a gene, okay, and you use a kit to randomly change any and every nucleotide. You will say, how I know? So in that case, you need a assay. What I mean by that, you have a DNA, you push it to any changes and put it into bacteria, allow it to grow. And the condition is, if it grow, this is a better variance. So in that way, these two works. Site directed, you know about this. Random mutagenesis, you don't know, you just take each and every conditions. Okay. So these two changes are coming as a procedure by the work of Francis Arnold. And this is what actually nature also is doing through evolution, right? Only thing is, we try to pick up our proteins which are needed and make quicker evolution than nature, right? Everyone, right? When Francis was doing this, computational biology was growing like anything. You probably all heard about AlphaFold, right? A lot of talking is happening. I'll talk about AlphaFold. But AlphaFold is coming. Now, let's say you have a protein. When I say you have a protein, how many amino acids that protein have, you think? Let's say you go to a Viva and I ask you, you have one protein. What is the molecular weight? Do you say, sir, you didn't tell me the number of amino acids because that is dependent? You might, but when in protein biochemistry we say you have a protein, we mean you have a protein of 300 amino acid. Why? From where this number come? If you take all the proteins and make a statistical plot, you get way high peak at the 250 to 350 region. So though in theory there would be a protein made of 50 amino acid and a protein made of 5000 amino acids, but the average amino acid is 250 to 350. Okay. Now let's say you have a 300 amino acid protein. How many amino acids are there? 20, right? The first position is methionine. You cannot change it, right? It is the first one. It have to be there for the protein to be biologically synthesized, right? Now you have 299 amino acid which could be changed to 20 possibility, right? The number of mutations, the number of variants of protein is 20 to the power 299. You know how big is this number? So I could say, if you go to your computer, take 11 font and Times New Roman, then it would be covering 75% of a page. Forget about the number, just imagine the volume of the numbers. And this is coming from one protein, right? So these numbers are called as sequence space. What is space? The volume which is covered by these numbers. Here we are not talking about the protein could fold or not, the protein could functionalize or not, anything. Just the number which could be created from a 300 amino acid protein. Everyone understand? Right? So this conformational space, let's imagine this whole room is the conformational space, like the sequence full of. Among them, what we are looking for? The ones which nature have given us. So I would say this box, black box, would be a very like overstatement that this is the number of or volume of protein nature is making if nature have probability of the entire room. So you understand how we are missing how many of them? And that was bothered a person called David Baker, who is this year's Nobel Prize winner. He came up with something. So this was the thing I was talking, was logical variance, logical engineering. Because you see something, you know about the functions, you do the changes. David Baker come up with something which is calling de novo engineering. What that means? That means he is trying to cover at least this many boxes, not a lot. Sorry. But that is much more than what is actually there with the nature. So how they are doing, what they are doing is important, but more important is this is opening a huge area. So I was talking about this protein, and the moment drug discovery is getting a lot more protein because of the Human Genome Project. So you see that this protein, it is deoxycytidine kinase. The name itself suggests that this protein works for deoxycytidine. Interestingly, it have broad specificity, so it could phosphorylate deoxyadenosine, deoxyguanosine in addition to deoxycytidine. So people thought that all the molecules we have are D molecule, right? What about the L molecule? So they tried molecule like 3-TC, L-deoxycytidine and all, and they got activity. So you see here a very interesting part of the slide where you see that the D-deoxycytidine and L-deoxycytidine both get the vicinity. What I mean by that, suppose you have this hydroxyl group to phosphorylate. Okay? So if this is the molecule, the hydroxyl group is here. This is D. The L would be like that. So hydroxyl group would be here. So there is much more space between them. So if a molecule do this, it is very difficult to do this. So that's why people thought the enzyme which is working on D-deoxycytidine could not be working to L-deoxycytidine. But when the engineering happens and the high resolution crystal structure came out, it shows that it's not. Why? Because chemistry will tell you the sugars used to be having a property of puckering. So because of that, both the D and L are not like that. They are puckering. And both of the hydroxyl group are now coming closer. So if an enzyme could hydroxylate D1, it could also hydroxylate L. So in that way, they could be hydroxylating compounds which are non-physiological that are used as nucleoside analog drug. So who are the nucleoside analog drug? So this is the one which we talked about. If you don't have this hydroxyl group and you could be incorporated, we all understand that there is no possibility to further elongate. So dideoxycytidine is a nucleoside analog, could be used as a drug. The next one is L. As we talked about, L nucleoside analog, they are little complicated concept because it's not straightforward, but we know about this puckering and all. So there is a possibility that they could also be working as drug. More interesting is this. By the way, if you don't know, there are lot of nucleoside analog, acyclovir, peniciclovir, ganciclovir, who are working for herpes virus, anti-herpes virus, antiviral therapy. How they work? Because you see that this part is open. So it could take any conformation, and luckily if one conformation goes and present the hydroxyl group to the active site of the enzyme, then it could be further phosphorylated, right? So then it could be used as a drug. So open chain sugar molecule, very interesting concept. I don't know if there is anybody who don't hear about remdesivir, right? We all hear news in time of COVID. So remdesivir, it's sold under the brand name Vacluri, and if you look at this molecule is also a nucleoside analog. Can you see that? It's more complicated, more other groups are attached, but it is also a nucleoside analog. So if I look about, you were talking about in time of drug discovery definition about the cost and all. So what is the process? The drug discovery process starts with target identification. Who are the target? We are coming. Then once you get the target, you try to incorporate small molecule to make some inhibitions or anything else. So that is called heat generation. Then once the heat is generated, you will make more and more more and more molecules to make optimization. That is called lead generation. Then once lead is generated, lead would be optimized, and finally you will get a clinical candidate. Everyone get it? So if I now do the cost assessment, target identification, heat refinement, lead refinement, and regulatory development like toxicity and all those things, clinical trials things. Obviously, the first one would be needing money, second one money would be added, third one money would be further added, money would keep adding. So if you look like that, it would apparently show you that the most costly or most important step is regulatory development. Though it is very important, but if you don't get the target identified properly, your whole drug discovery process will not be proceeding in a proper direction. So, yeah. Someone asking anything? Okay. So, target identification, validation, chemical libraries, combichem, natural products, all are going through the high throughput screening to develop the lead compounds. And who are the targets? Lipids as cell membrane lipids, proteins as receptors, enzymes, carrier proteins, structure proteins, nucleic acids, carbohydrates, all could be targets. But protein is always the most important target. Why? We will talk about. But before that, because it's the first session, I will make a summary. So drug discovery is creating small change in chemistry which greatly affects a biological phenomena. We have understand this by the example of nucleoside analogs as drug candidate. We have also understand cost assessment in the drug discovery process. Our understanding clearly suggests target identification is the key to drug discovery, and most important biological target is biological macromolecule called protein. So, coming back to biology, to understand biology, cellular function is an organized demonstration of millions of molecules. And for normal functioning of the cell, it is extremely critical that these molecules communicate properly. What is biological communication? Let's imagine, you don't have to imagine because I am really drinking some water, right? while I am drinking some water, let's imagine some of the one drop of the water fell on this step bench, right? Was possible? And one poor bacteria was just passing in through this bench. Now, what will happen? To that bacteria, suddenly there would be a sea of water created, right? Is it happened to you when you are coming here? Is it happened that you don't know but suddenly a river created? No. That's what the property of the microorganisms, right? So, you know, when Human Genome Project happened, all people know about before that through investigation of E. coli. And people thought they know E. coli very nicely. But when Human Genome Project leads to all these, you know, high throughput and all the bacteria were like genomes were sequenced, scientists were very surprised to see that 1/3 protein of bacteria is what we don't know about. Most of them coming in a category called two-component system. You know about two-component system? Anyone? So two-component system is a signal transduction system in bacteria through which bacteria communicate in different situations. Okay. Two-component system is very interesting. In two-component system, there is a histidine kinase. When I say histidine kinase, how many students think something about that? Like by the name, what you understand when I say histidine kinase? How many of you have biology background? At least have biology. None of you? Very few. Sandeepan told me a little different. Okay. So, histidine, when I say histidine kinase, that means it is a kinase, it take part in a group transfer reaction and it use histidine as a catalytic molecule. Like for the enzyme, there would be a catalytic residue, so where the group transfer would be happening, phosphorylation will happening, this is histidine. People who are in like from biology, they know probably there are 3 known kinases who are phosphorylated: serine, threonine and tyrosine. Now if you look at the chemistry of serine, threonine and tyrosine, all of them have a hydroxyl group. Okay, that hydroxyl group is phosphorylated. Okay, why I make this background? Now you see, you go back and check the amino acid of histidine, you will not find any hydroxyl group in the side chain. Okay, so that's where the interesting thing began. Generally, if you look at, if as I see very few hands are from biology, then rest are from chemistry, now you could have easily understand that there was a OP bond developed, and now histidine in its side chain have nitrogen, so there would be a NP bond going to develop. What is the difference? The OP, because of the difference in the electronegativity, would develop a relatively stable bond than NP, right? So remember that in next round we will talk about that. Okay. So histidine kinase, it have one domain outside the cell, rest of the domain inside the cell. Okay. When the so when the like cell is coming, they used to be phosphorylated. Now when you look at that, you see that most of those histidine kinase, they have a common structural motif. So this structural motif is important for their dimerization. That's how we could understand by the study of, through structural biology, how it is functioning. This is the part you could see in every different histidine kinases, right? And these are the functional domain which is doing the catalysis. Okay. Now you could see that it dimerizes now, phosphorylate, but phosphorylated in a molecular level, it is phosphorylated through NP bond, right, which is much less stable. So it would be trying to pass that phosphate, right? To take that phosphate, here is coming the response regulator. These, so we had one component histidine kinase, another component response regulator, so it is called two component system. One thing. Second, the residue phosphorylated here is not even a serine, threonine or tyrosine. It is a aspartate. So people who are again not knowing about these amino acids, this is a amino acid with a acid group CH2COOH in the side chain. So it does not have a direct hydroxyl group but have a resonance stabilized hydroxyl group. So now if I compare, there are three group of hydroxylated molecules. One is OH, which is in the middle. One is very unstable, which is N, and one is very stable, which is resonance stabilized OH, right? Now in bacteria, Mother Nature is testing it, testing this model. Why it works? Because there is a potential difference from very unstable to very stable, so the phosphate easily coming here and the running the further prospects. But if you see in biological system, there are series of phosphorylation needed. So it's not one phosphorylation in our biological complex biology system. So if you are throwing something from the top, niche, it will be going easily, right? But if it is a series, you cannot do that. So these two component system, they don't exist in higher organisms. So this is a very interesting drug target, and nowadays you guys have heard that we are really low in our arsenal because most of the antibiotics we have created are under resistance. These are giving us new hope, these are giving us new targets. But what about our story? In our story, it's coming here, and now when it comes here, it goes to further expand their signals by DNA binding, RNA binding, protein binding, and small molecule binding. So there are each and every way they utilize to further doing the signal, further expanding the signal. So they get a signal and they further downstream that to the bacteria to know about what is happening. Now this DNA binding is also very interesting. You see here, there are different families like Narel family, Phoebe family, NRRC family, Aracy family, Fish family, all of them are DNA binding, all of them are two component system. But if you see, the symmetry of binding is very different. And those symmetries are very important for signal. How tightly they are binding, how, depending on how the binding affinity is more or how the binding affinity is less, all of them are affecting the biological process. So, and in that way, they do the communication. So, two things here, we try to understand the communication process. At the same time, we see how this biological structure and their chemistry is very, very, critical for these communications, right? So, in biology, communication would be mostly performed through interaction. To understand the process of communication, we need to visualize molecules at atomic level. We need to determine the proper arrangement of the atoms in those systems, as we see that how the dimer is forming, how the catalytic residues are there, how the ligand is there to, like ligand binding site is there to get a ligand. All of them are dependent on that. But there are different level of interactions as well as size of macromolecules, or their organizations are also quite different. And based on those requirements, there are choices as well as advancement of the techniques. So, who are the techniques? Among the high resolution techniques, there are macromolecular crystallography, X-ray, there is nuclear magnetic resonance, NMR, cryo-electron microscopy, or cryo-EM. So, how size dependency is there? I have given a brief comparison of a body starting from the tissues to going to the biological macromolecules, where we look at a atomic scale. Now, from 1 millimeter to 0.1 nanometer, you see that tissue, then cell, then organelles, then biological macromolecules. At the initial stage, you could see them using light microscopy, but then when you go like even deeper, even smaller side, at the bigger level, among these smaller things, you get cryo-electron microscopy. Cryo-electron microscopy cover nearly from 100 nanometer to 1 nanometer. Excellent range. Then come macromolecular crystallography. Much bigger ones are not acceptable, but from there it also contains a good range. Then comes NMR, which have quite a small range, because NMR in protein is not possible for bigger protein molecules, because I will talk about that in details, but NMR depends on the movement. Higher the molecular size, the tumbling become slower, and because of that the spectra start overlapping. So now with the best conformation of the NMR, which is 1.2 gigahertz, NMR is talked about in terms of the magnet, external magnet. So you see that here it is possible to work maximum up to 80 kilodalton, 90 kilodalton proteins. But there are many low resolution techniques which we are not going to discuss today, but I want to just talk about circular dichroism. Circular dichroism determine the ellipticity, and from ellipticity you could see that how the proteins fold properly or not. You could also study a lot of protein drug interaction using circular dichroism. Fluorescence spectroscopy works in two ways. In one way, there is a intrinsic fluorophore. What is the intrinsic fluorophore? So inside the protein, there is a amino acid called tryptophan. Tryptophan shows high quantum yield and fluorescence. So by taking tryptophan, you could study the movement and folding of the protein. But in addition to that, you could also add external fluorophore to study a protein. Electron paramagnetic resonance is a very interesting concept if you know about NMR. How NMR works with odd number of nuclei, EPR works with odd number of oxidation state. For example, zinc have 0 and +2. For zinc, EPR will not work. For metals which have odd number of oxidation states, only for them EPR will work. Small angle X-ray diffraction or SAX, it work at relatively lower resolution, but it looks at the movement of the molecule. It creates the dynamic data, how the molecules are coming together, how they are interacting and all in a relatively low resolution. Isothermal calorimetry, it is one of the finest process to measure protein drug interaction. It creates a lot of thermodynamic parameters, like it measures the enthalpic change del H, it measures del S, the entropic change. Also it measures N. N is the stoichiometry. Why stoichiometry is important? A lot of time we see that a drug is binding to a protein, but what mole of drug binding to 1 mole of protein, that we don't consider, and that mislead us in many protein drug interaction. ITC, isothermal calorimetry, tells you the exact proportion how a drug molecule interact with 1 mole of the protein target. And beside that, we know about light microscopy being low resolution, but fluorescence microscopy, confocal microscopy provide us a lot of low resolution critical data. So why we need 3D structure of protein? As we talked a lot, but if you look at among the macromolecules, DNA, RNA and protein, protein is the functional molecule in biology. So that's why it is more critical in functional purposes. And if you look at the protein fold, you will realize that by looking at the fold of the protein, you could guess the function in a certain way. And in other molecules like DNA, RNA, by knowing the sequence, you could guess about the function a lot than you could understand in a protein without knowing the 3D structure. So there is a beautiful correlation between structure and the function in a protein. So when we are talking about, we now talk about to read the molecule. So before that, if you don't know about that, this is the whole cycle of life where DNA produce DNA through replication, DNA produce RNA through transcription, and RNA produce protein to translation. This thing is basic to a life, and it is called the central dogma of life. Now, with the sensitive machines coming, we are not only targeting that, but also we target the sugar, nucleotide, amino acid and lipids, because those molecules also tells us about the function. if you compare between a normal body and a diseased body, these molecules are very good indicator. So how we could reach there? So first we have to know what are the factors, what are the total molecules which are there. The whole DNA content of a cell is called genome. The whole mRNA content of a cell is called transcriptome. The whole protein content of the cell is called proteome, and the molecules generated through the action of enzymes are called metabolome. So you see in that series, who is at the top? The DNA, right? So if you could get DNA, you could get all of them. How? So when you get the DNA, through that codon relation you get the, so they are the same, both are nucleosides, so you get DNA to RNA, so you get the genome and transcriptome. Then you come to the protein, amino acids, and then from there if you follow up the metabolic pathway, you get the metabolome. Basically what I mean, by getting the DNA content, it is possible to further figure out all the other informations. So that leads to something called Human Genome Project, and as I told, Human Genome Project was initiated because few reasons. One of that, people want to think that like before human genome the project happened, like before people have sequenced a human genome, all of them thought that, and there was information among scientists that between a chimpanzee and a human there is less than 1% difference. Okay, so everyone thought that, you know, I don't need to do it, right? In US they are thinking like Trump will do it. Here we think Ambani, Adani will do it, right? So that was the situation. Everyone thought it is less than 1% with chimpanzee, and I would have better, you know, homology with Adani, Ambani and Trump. So if they will do, we will get our sequences, right? That was the idea when Human Genome Project was not happened. That idea changed hugely after Human Genome Project happened, and that is a kind of revolution in biology. You know, when you go in the course and sit down in the subjects, if you look at the history, most of the history started 1800 and all, and most of the innovations are done at 1950s. Not this. Human Genome Project finished in 2003, but the effect is coming to exactly in your generation. Why? Because in 2003 it came into the market, but the price was way high. I am giving one example. I joined, as Sandeepan told, 2014 I came back to India, joined IIT Roorkee and take my first quote for bacterial genome, because I work on antimicrobial resistance, so we do genome sequencing of the bacteria. And that time GST was 5%. So with GST 5%, I get a quote of 1,60,000 for one bacterial genome sequencing. Cut to today, I pay 15,000 for the same with 18% GST. So there is kind of around 11 times lower. So within 10 years, in place of, in the same money, spending the same money, in place of one bacterial genome, you are getting 11 bacterial genome. The informations are coming hugely, and you know that more information in your world. There is AI, there is ML, there is data science. So this is what your subject is. Exactly now this thing is growing. So let's talk little bit about Human Genome Project. So to read, we call sequence, right? So to know the sequence in molecular level, what is the trick? The trick is, and that's why I started with nucleoside analog. I know I have to come here. So you already know that if I take out these ones, what will happen? Remember nucleoside analogs, chain elongation will not happen, remember? So that's what I wanted, you all remember that. So now imagine if you create those molecules, a molecule like this, dideoxy we talked about, right? What will happen now? If you add this and do a DNA sequencing, the DNA sequencing will be stopped. Everyone agree? But now if you keep continuing, then you get some fragments, right? Tick, because it will randomly incorporate, you will mix, there will be a competition between the nucleosides and nucleoside analogs, where nucleoside analogs will entry, stop. So you will get this much, this much, this much, all the fragments, right? What I am talking about, this is the concept of someone called Sanger, and this is famous as Sanger sequencing. But why I am not totally talking about Sanger sequencing? Because I have to talk about another thing. What is the another thing? Polymeric chain reaction, or PCR. The time Sanger is coming up with the concept of this nucleoside analog, at the same parallel time Carey Mullins coming up with a machine which could synthesize DNA outside the body. Okay? Only what was the challenge here? The enzyme to work at higher temperature, and they solve it with a thermostable enzyme, and it happens. So though I am talking now about Sanger, it is a joint effort of Sanger's idea along with Carey Mullins instrument. What happened? The DNA is fragmented. Why it is fragmented? Because Sanger sequencing cannot do all the DNA. It have to do the reaction, right? So the DNA is fragmented to make it smaller. You get the smaller ones, that is the template, because DNA needs a template to synthesize, right? So you give that, and then you give the nucleoside triphosphates, DNTP along with DDNTP. Okay? So there would be a competition. Initially, they detect it with a big plate by radioactive labeling, which is very risky. But then what happened? This human genome sequence initiative started. Why human genome sequencing initiative is important? Because through this human genome sequencing initiative, big universities, name them MIT, Harvard, Oxford, Caltech, all are coming in the one base. Okay? When they are coming into one base, though there was no business apparently, but companies thought that if they come, they will at least hit the newspaper, they will get popularity. You understand? So big universities develop a platform to sequence the human genome. Why? Because that time you have to manually read. Like in your PhD, after 1 year work, you read ATGC, this small, with 3-4 years of work. That was the reality. People used to read one gene, two gene in an entire PhD. Okay. Suddenly what happened? They start making this platform. Biologists meet technicians. They told, why you are making radio level? We have so beautiful fluorescent level nowadays. Immediate change. This fluorescence labeling. Sensitivity suddenly increased 1,000 times. They were running these big gels. Engineers told, why? Run capillary. You get much higher resolution. They start doing capillary. And through capillary, now they could read. They could separate two nucleotides. They have this fluorescence level now, so they could fire with laser and they start live reading. So they don't need to do the sequence and then reading. It's the live one. It's going, and there is something called chromatogram started developing. So in one moment, what one PhD student was doing in 3-4 years suddenly become a matter of 3-4 days. After that, 3-4 hours. Now, 3-4 minutes. So that's how the technology and biology comes together, and they develop this Sanger sequencing. They used to sequence 800 plus nucleotides. If you don't have idea, I talk about protein. 300 amino acid is the average protein. 300 amino acid means 900 nucleotide. Okay? So 800 nucleotide base length read, accuracy approaches 100%. In which year? In the year of 1980s, they appear actually 100%. And even today, Sanger sequencing is considered as gold standard. So we could see now, we could see the genes now, but we want to see more. We want to see the genome. We know that every human's mystery is written in their genome. So the good part of human being is we are not happy with anything. We get something, we want to go higher. So Sanger sequencing is the first generation sequencing, and then further when we go for parallel read to read the gene to genome, this is called next generation sequencing or NGS. So why NGS? The basic characteristics: generation of many millions of short reads in parallel. The speed up of the sequencing process compared to the first generation. The low cost of sequencing, and the sequencing output is directly detected without the need for the electrophoresis. So Human Genome Project 1990 to 2003, people consider this as a small step for man to get immortality. It was published in Nature, Science, big journals, and it was considered so important that the then President of America, Senior George Bush, came and collected the entire human genome data in 12 CD-ROM. What is the effect of NGS? The price start going down. The genetic engineering is becoming possible, and even before birth, like the genetic defect which are known could be rectified. So what is the workflow? I am going quickly. Now, genome DNA. So first you have to select which genome, my genome, your genome, you have to select it. Then you have to construct a library. Why you need to construct a library? Because first I would break them, but then I have to read that, I have to reassemble that. So that's why we have to create a library, prepare the template, and then we will do massive parallel sequencing and ultimately the analysis of the data. So these are the instrumentations. As I told, the first generation is ABI Sanger, Rose, then is the first generation, then second generation, then PacBio and Illumina is the third generation, and Oxford Nanopore is the fourth generation or last generation. As you see, if you like, those guys are much expensive. If you look at this one, this is only 1 lakh rupees cost, and you could hold it like that as it is showing. So application, it have universal application. This is one of the best slide. You could get any and every information in the space, in the water, tissues, inside the body, anywhere, if you get a piece of DNA. So that's the power of NGS. We use it in antimicrobial resistance. You could, track people who are coming in India or going from India, how this is spreading and all, you could have all track. So overall effect, as I was talking, before that you collect really small data. This girl is showing her data. Upper panel is my 4 years of work, and the lower panel took only 1 year, which means it's a lot more time and very less data. Then suddenly, things are talking big, and the departments where there was department of biochemistry, genetics, who are working on one gene, one protein, suddenly there is big whole things, proteomics, genomics and all. A scientist, when asked that, what you think is the successful term for yours, he answered that, when I can coin my own OM word, genome, proteome, transcriptome, OM was all over. And as I told, the price reduces a lot, so it even affects God. So when God knows that this human genome project was done, an angel came and told the God, the human genome code been unraveled. God is also, very like scared. Damn hackers, now I have to change the password. So it was so impactful that even God was affected by that. After genome sequencing project, now we have millions and billions of gene sequences, which means we have that many protein sequences known, but we need to know the 3D structures. So God is now getting confidence back. Perhaps I don't need to change the password. They still have a dynamic proteome code to break. They cannot hit a moving target. So challenge of protein folding, this is the significance in the field of biological research. What is the importance of a folded protein structure, how we could determine the protein's 3D structure, and what are the level of different structures where we are working. So this is the first part. In the second part, I will talk about the structural biology techniques, but you could take a break. 5 minutes, we'll come back. Thank you. But I will be more happy if you guys come front and ask me more questions. শিবাশিস ওইটা একটু সেট করে দিও, আর আমি রেস্টরুম যাচ্ছি।
Participants who ever did not fill up the Google form, please fill up that as soon as possible. Google form. Google form. You have to, yeah.
ठीक. So I'll go to the next part. So we are talking about structural biology techniques to start with steps in protein crystallography. As I told, in protein crystallography or any of the structural biology technique, the most important part of the process is to identify the target protein, and then you want to produce that because it's needed to be found in very pure form. So first what you do, you have to ensure that you could purify it to the good amount of protein you could get. So we do cloning. This is a genetic engineering technique through which we get the protein separately in a high amount. After that we go for overexpression, which means the bacteria the bacteria have, let's say we are taking E. coli. E. coli have 5,000 other proteins. In between, your protein is the 5,001 protein. If it produce 1 by 5,001, you would not be happy with that. Your money would not be properly utilized. So you want somehow to do something. I'm not going into them. There are many overexpression techniques by which you want your protein to be growing at least 80%. Once it is growing overexpressed, you would purify it. For crystallography, the protein purification is very important. Why? Because the process of crystallography is to change a phase from liquid phase to solid phase, and the protein have to be very, very homogeneous. That's why the proteins needed to be very, very pure. When I say very, very pure, I mean at least 99% pure protein is needed for getting crystal. Then you go for crystallization. Crystallization is the step where everybody is stuck. There is no science in crystallization. We just keep setting up crystals and pray for getting a crystal to come. Once a crystal is coming, we then go for mount the crystal, diffract it. Then one of the problem is there, when you get the electron density data, you get intensity but you don't get phase. That is the classic phase problem in crystallography. And then you do the model building, then you do refinement and data submission to protein data bank. Protein data bank is now very serious about data submission. Why? Because if you go 20 years earlier, the data is utilized to look at, right, to analyze. But now a protein data means it's a PDB file. A PDB file now is taken by computational biologists too. So imagine you are a crystallographer, you make a PDB file with 10% error. Now a computational biologist have no idea about that. Your paper is published, your structure is in PDB, he or she will picking up that. So they are starting with 10% error. That's why in 2014 there was meeting, and two meeting concurrent at 2012 and 2014. After that, the PDB working as a database, now they are working as a proper data bank, where I am a crystallographer, so a scientist like me is there, sitting there. When I am giving my data, he or she is going through the data, and if there is errors, he or she write there and send back to me. I have to again work on that, correct that, and only after that I get a validation certificate. After getting this validation certificate, only then you could mention it in a publication which you are sending that I have a crystal structure solved. So the process is more rigorous, which means the process is more corrected now. So just few of the things. Why cannot we visualize molecule directly using X-ray? A single molecule is very weak scatterer of X-rays, right? Most of the X-rays will pass through the molecule without being diffracted. The diffracted rays are too weak to be detected. So in crystal, we don't have a single protein, we have a array of millions of proteins. Think about, if there is only one person, it is very hard to see. But now I could see all of you because a lot of you are there, right? It's same. You, like single U is a single protein, all of you is a crystal setup. Okay. Solution, analyzing diffraction from crystals instead of single molecules. A crystal is made of three-dimensional repeat of ordered molecules 10 to the power 14 whose signals reinforce each other. The resulting diffracted rays are strong enough to be detected. So I will proceed, but here I would love to talk about small time I will take to mention a technique. You know, you all know about Raman, right? Raman is a very convenient technique. Raman work as a confirmatory experiment, right? Right? In protein also, Raman detect a lot of fingerprints. Fingerprints means you get those, you know that this group is there, right? So Raman could be a very good asset for studying protein, but anyone who know about or who read about protein, or if you go back and study about in the literature, you will see that Raman is not used for studying protein. Why? Because Raman gives very faint signal, you know that. That's why most of the Ramans are either surface enhance or resonance, right? FTIR and all those things coupled with. So that's the problem with protein. When you add laser, what happens? There is generation of heat and your protein will lose the fold. Okay, if it lose the fold, you cannot study the function. That is the reason, in spite of being a very good technique, Raman is not used to study protein. Okay. So I was lucky enough, when I was in US, I met a guy, William Perry. William Perry and group, they develop a technique called Raman crystallography. What is the technique? The technique is simple. You know confocal microscopy, where you could measure the Z, the width, right? So in Raman crystallography, there is a confocal. But in confocal, from where the source come, light, right? Instead of light source, it is changed with a Raman monochromator. Okay? And there is a very high resolution CCD camera. How high? I could just tell you the price. The price is around 1 crore for the camera. Now I am coming why it is very important and why it's like people are following it. So now you are not setting up a protein solution. Instead you set a protein crystal. You already see that 10 to the power 14, right? Protein is 10 to the power 14 protein molecules are there in the crystal. So crystal is used there, and from the confocal, the Z, the 3D axis is focused, the pinhole is focused on the crystal. So the machine don't take any signal out of the platform of the crystal. You understand? So inside the crystal, the jade, this is only focused. So if there is solution, there will be no data coming from them. It will only acquire data from the crystal. Now you do reactions. Okay, you add a molecule which would be reacted with the protein. The molecule before adding, you could take a Raman, that is the baseline Raman, and now each femtosecond you start collecting the data. If there is any change, the baseline would be affected. You see a change. Are you getting me? So you could actually see the reaction in a femtosecond time level. Obviously, you don't know what is happening, but later that signal would be quantum calculated and you get the structure of the molecule or the chemistry of the molecule. So you are not looking at the protein, but you are looking at the enzymatic effect which is coming on the drug molecule or substrate molecule on which the reaction is happening. You understand? So that instrument is called Raman crystallography or Raman confocal crystallography. Coming back to, unlike visible light, X-rays cannot be focused by lenses. The refractive index of X-ray in all material is very close to 1. Solution, you don't need to use it directly, use a computer to simulate an image reconstruction lens. So instead of lens, you are using a computer to simulate. The computer plays the part of the objective lens, computing the image of the object, then displaying it on a screen. The nature of crystal, under certain circumstances, macromolecules, protein, DNA, RNA can form crystals. The resulting crystal is a three-dimensional array of ordered molecules held together by non-covalent interactions. So this is a crystal, that is a crystal, but these first three are inorganic crystals, or the last one is a ice crystal. The fourth one is a protein crystal. So evidence that solution and crystal structures are similar. So initially it used to think that you are making a crystal. Is that crystal behave like the solution behavior of a protein? So if you ask me, obviously not exactly. So I am moving, you are sitting, there is a difference between our movements, right? So similarly, when it is arrested, its movement is restricted. But as people have thought that when a protein is going in the crystal, its activity is totally lost. Come to me, fortunately, our work is with a protein. You know, protein is a catalyst, right? So protein don't directly react with a substrate or don't create a bond, right? We work with a family of enzyme called beta-lactamase, which work with beta-lactam, the penicillin stories. So that enzyme form a covalent bond with a substrate. Why I am talking about that? To tell you my research? No. I am trying to tell you, so when my protein is, so if a protein is developing a non-covalent interaction, you cannot prove that it is happening inside the crystal or not, right? But if the protein form a covalent bond, then definitely you could say that my protein is active. So we form crystals in the apoform. What I mean by apoform? When the protein is only there. Okay? So we take the pure protein and set crystal. We get the crystal, we soak that crystal with the drugs, with the antibiotics. And what we get, you know, if you look at penicillin and all these beta-lactam drug, there is a 4-membered ring. We see the 4-membered ring is broken there and it form a covalent bond with the protein. That means our protein is active inside the crystal, right? So there are many such proofs coming out now. So it is not that proteins are not active there. But the scenario was actually like reverse, because crystallography, the first structure came at 1959, Kendrew and Perus, right? But NMR came much later. So this is the structure 2ZZZ2 is a structure of a same protein. You see 3C4S. 3C4S was solved by X-ray. But when first structure was solved by NMR, this was the first structure solved by NMR, but to prove that it is correct structure, they had to crystallize it. You understand what I mean? So because X-ray was more established technique, to establish NMR, they solved the structure with NMR, but they had to show that they are same. But at the same time, it proves that the structures which are in solution, the structure which are in X-ray are same structure. Many macromolecules are still functional in the crystalline state, and most protein crystals contain 50 to 70% solvent. So if you are first one, go pick up the crystals, you are the first one, so your hand is like this, trembling, being tensed, and if a small touch goes there, it might spoil a crystal. So the first thing we teach our students, don't be happy with one crystal. If you get one crystal, try to make at least 100 so that you could spoil 99. The ideal crystal, which is not a protein crystal, the ordered disposition of molecules such that over a distance equal to or greater than thousands of molecular dimension. The real crystal, a crystal with less than perfect periodicity, imperfections are often caused by impurities and the effects of non-zero temperature. The protein crystal, the crystal contain a high degree of solvent, meaning that some molecules present are not in crystalline state but in the liquid state, creating disorder. These are the examples of some of the beautiful crystals which are very less common to be appear in the lab. Process details of protein crystallography, as I told, we first do the cloning, we do overexpression, then we get purified protein. You see a very strong single band is required. Mostly you have to do 2 or 3 stage of purification. Generally we do 2 stage of purification. First we do affinity chromatography because our protein have a tag. When we clone, we incorporate a tag of histidine. That histidine tag is bind to the nickel NTA and purified by higher imidazole. So that's the first purification. Second purification is size exclusion. There are bits, proteins have to be going inside the bits. The second one, the size exclusion, is very, very important for protein crystallography. Why? Because sometimes proteins are, you see protein fold, but the protein is not properly folded. Okay? It is like locally denatured. When you do a size exclusion, it will go through the bits, it will be properly folded. Okay? So that's why size exclusion is very important. Then we go for crystallization, we get crystal, we get diffraction, and then once we get diffraction, we go for solution, we get this electron density, and from the electron density we get the structure. So what we need for a good crystallography, need to have the protein in a very pure state, need protein to crystallize, need very good crystal, need good diffraction, good resolution. There is very interesting recipe of crystallization. What we do, we make a thermodynamically closed system. How? We take a well and a reservoir and take a precipitant, because our protein is in liquid state, right? And we want to get it solid. How a protein could be solidified? Only by, do we want precipitate? No. That's the fallacy of crystallization process. We push it to a situation what we don't want. Why? So it is liquid, we push it to precipitate. So there is a change in the phase diagram, right? While it is changing, there is a most elite point. If it stuck that point, it will crystallize. How much is the chance? If you do everything correctly, there is a chance of 8% to get a crystal. But when I say that, there is nothing to be disappointed, because when you are working with your protein, first of all, in crystallography lab, we work with multiple proteins, and when you work with your protein, you know your protein a lot. So you do a lot of mutations to change the surface property of the protein so that the protein could be crystallized. So here what is happening, see, you have the precipitant, you have the protein. You take 2 microliter of the precipitant, which is in the reservoir, and 2 microliter of the protein. What will happen now? The reservoir solution is half diluted here, correct? Now what you will do, you take it and close the reservoir, making a thermodynamically closed system. Okay? But in the drop, the reservoir concentration is half. So from there, slowly water would be flowing, right? And gradually the protein would be concentrated. More the protein would be concentrated, more it is going towards precipitation. It is lucky, as I told, it would be crystallized or it would be precipitated. So most of the time we get precipitation. So should make large crystals in less than 15 minutes to few days to even months. I got crystal after 1 year. I see people got it in 10 minutes. So this is because of creation of supersaturation. So to add more of a substance to a solution than can normally be dissolved, this is a thermodynamically unstable state achieved most often in protein crystallography by vapor diffusion or slow evaporation techniques. So as I was talking, you have protein and you have the salt or the precipitant. Now you have 3 zones, zone 1, zone 2 and zone 3. So zone 1 is the metastable zone. The solution may not nucleate for a long time, but this zone will sustain growth. It is frequently necessary to add a seed crystal. So what is this situation? You set up, you keep checking and nothing is happening. So what you will do, you see that your friend got crystal. It's another protein, no relation with your protein, not a matter. Take some of the crystals, okay? Take it in a pen drop, vortex it. So your crystal would be breaking into many nucleation steps, like fragments. Then put a cat whisker. You know what is a cat whisker? Take your hair, okay? Put your hair there. Whatever is coming by touching the hair, touch the drops. They would be nucleated. They need 1 nucleation. Zone 2, nucleation zone, protein crystal nucleate and grow. Zone 3, precipitation zone, proteins do not nucleate but precipitate out of solution. So nucleation, it is what we all want. It is a phenomenon whereby a nucleus such as a dust particle, a tiny seed crystal, or commonly in protein crystallography, a small protein aggregate starts a crystallization process. Common challenges: if supersaturation is too high, too many nuclei form, hence an overabundance of tiny crystal. What to do? Again, pick from there, dilute it. In supersaturated solution that don't experience spontaneous nucleation, crystal growth often only occurs in the presence of added nuclei or seeds. So this is a unit cell. When you are solving, you are getting a unit cell, and the crystal would be growing around the unit cell. It's a repeated process of the unit cell and going in 3 dimensions. So obtaining suitable single crystal is the least understood step in X-ray crystallograph structural analysis of a protein. Protein crystallization is mainly a trial and error procedure in which the protein is slowly precipitated from its solution. Protein crystallization is an art than science. What are the important factors which can affect formation of protein crystals? So factors इसको एक बार रीस्टार्ट कर देना. Thank you. So factors that affect crystallization: purity of protein, protein concentration, starting condition, makeup of the protein solution, precipitating agent, temperature, pH, additives like detergent, reducing agents, substrates, cofactors, homogeneity of the crystallization solution. And while I am talking about, remember I talked about Human Genome Project. So when Human Genome Project got success, and the whole credit goes to kind of to US, they come up with something which is called structural genomics. The concept was, like in the Human Genome Project, they started with a human, but then that process keep like making genome of other organisms. Here also they take one organism, they take all the genes, they take all the proteins from there, produce all the protein, clone, then overexpress, purify, crystallize, and if possible, crystallize everything. That project they call structural genomics. So the success of NGS based human genome sequencing opens up the process of high throughput genome sequencing projects. Millions of genes were sequenced, hence millions of protein sequences are known, but to proceed further, we need the structure. To fill up the gap, there was a development of an initiative by Protein Structure Initiative of National Institute of Health, US, called Structural Genomics Consortium to determine the three-dimensional structures of medicinally, health-related and biotechnologically important proteins. The Structural Genomics Consortium aimed to places all its research output into the public domain without restriction and does not file for patents and continues to promote open science. So there are different structural genomics centers, NYSGRC, which is New York Structural Genomics Research Consortium, NESG Northeast Structural Genomics Center, MCSG Midwest Center of Structural Genomics, JCSG Joint Center of Structural Genomics, CSMP and GPCR are for membrane protein structure solution. Goal of these structural genomics research consortiums were classifying all available genomic sequences to establish a prioritized target set, like who are more important proteins, take them earlier. Cloning and expressing genes and gene fragments of microbial and eukaryotic origin, purifying and crystallizing native and derived protein for X-ray crystallography, collecting data and determining structures and analyzing the structures for fold and function determination. So remember I told you that crystallography is special because crystallization, right? So I just want to show you an analysis of one of the structural genomics data and a reason where structural genomics was not successful. So the data is here, Joint Center for Structural Genomics, which was established at 2000, okay, and the data is 10 years data, so 2000 to 2010. Selected targets 33,209 genes were selected. Cloned 27,959 were cloned. Expressed 27,640, which are giving protein. Crystallized 2,128. Solved 145, deposited 968. So now if you see percentage from selected target to cloning, there is a success rate of 84%. From cloning to expression, there is a success rate of 99%. From expression to crystallization, 8%. From crystallization to solved, 49%, and from solved to deposited, 93%. Coincidentally, the number 8% actually matches with the statistics we generally talk about general crystallization possibilities. It's not that we are looking at these statistics. It was there before. But interestingly, you see, unlike Human Genome Project or any project, here the bottleneck comes when the crystallization is happening. So, growing a suitable crystal is such a hurdle. But why it is so hurdle, right? Why it is such a difficult process? So in a crystal, the diffraction signal is amplified by the large number of repeating units which we talked about. Diffraction from a single molecule is not currently measurable, whereas a 100 millimeter cube crystal contain 10 to the power 12 unit cells. So diffraction is much more. Diffraction intensity is proportional to the number of unit cells in the crystal according to the Darwin formula. And if you see here in the crystal, you see how the 3 dimension is developed, right? when a crystal is ordered, strong diffraction result from constructive interference of photons. What it means, your crystal cannot be developing in an unordered way. So what about the interference being constructive? Interference is constructive because path lengths differ from some integral multiple of the wavelength. Okay, this solution is possible only because the diffracting objects are in periodic relation. So only then you get the diffraction. So what if it is not? So you see that here this is a perfect order, right? Here there is a rotational disorder. Here translational movement. So disorder destroys the periodicity, leading to streaky, weak, fuzzy diffractions. And now you see these are the diffractions. You see this is coming from perfect one and the spots are perfect, right? Whereas when it is rotational disorder, you see the spots are stretched, right? Also in translational, it's stretched in a different way. So what makes crystallization such a difficult challenge? So we try to look at in a thermodynamic way. We all know that del G equal to del H minus T del S, right? And del G should be negative, right? So we are looking at thermodynamics of the growth of the crystal. So del G crystal equal to del H crystal minus T del S of the protein plus del S of the solvent. So we imagine this as protein in solution and protein in crystal. Is the del H crystal favorable? So if you see, yes, del H crystal is modestly favorable, 0 to minus 17 kilocalorie per mole. Here the protein in solution, here the protein is in crystal, the crystal lattice contacts are having large area, specific and rigid. Is del T del S protein is favorable? This is protein in solution and this is protein in crystal. So the protein in solution is moving, right? So protein in solution, there is 3 degrees of freedom in orientation, 3 degrees of freedom in translation. When it is moving, the entropy change is favorable, right? So here in case of like T del S, the protein is in solution and the protein crystal is not in movement, right? So this is having higher entropy, this is having lower entropy. So the change of entropy is strongly unfavorable, around plus 7 to plus 25 kilocalorie per mole. So here there is 0 degrees of freedom in both the cases. Is del T del S solvent favorable? Because here actually you see opposite thing is happening. They are moving, but the water molecule is bound to them. So the water molecule is actually not moving. Whereas here the proteins are not moving, so the water molecules got freed and they are in movement. That makes T del S solvent favorable. So the overall effect del G crystal equal to del H crystal minus, it gives del G crystal a negative but very small negative. So as I was talking, why they fell, why the bigger project fell and why the smaller lab like ours are running. Because when you are doing high throughput work, you cannot put attention to a single protein. Like if I ask you care of 100 protein, you will try to do in a high throughput way. But if I give you 1 or 2 protein, you will try to understand the protein, right? That's what the difference between a structural genomics project and a small lab where one PhD student is giving his or her life to get a crystal. There are many strategies. I will not spend a lot of time, but just like if you eliminate the floppy mobile termini, crystallization means getting rigidity. More rigid your protein, higher chance of getting crystal. So you take out that and you get the crystal. Similarly, you express, you have 2 domains connected with, you cut them and grow separately. So that works. Then you add a ligand between them. There are 2 domains, you add a ligand, that ligand stabilizes them. You get it mutated. You do mutations. For mutations which makes the surface more ordered or interactive, you get crystals. Serine, cysteine to serine mutations, serines are hydroxyl group, they are doing small interactions, stabilizing the surface and getting, like helping you to get the crystal. So increase the protein to favor crystallization. How you go from monomer to the crystal? To crystallize a protein, you need to increase its concentration to exceed its solubility. So how we do that? Again, something you could do when you are taking care of 1 protein. Something is not possible when you are taking care of 100 protein. You have 1 protein, you purify the protein, you check its individual solubility and increase the solubility. So force the monomer out of the solution and into the crystal. That is the condition called supersaturation. Now we talk about human genome, but if we look at the human genome, what was the first genome to be sequenced in a human genome project? Is the human genome. When was the first human genome sequence complete? 2003. How long it takes? 13 years. And how much did it cost? $2.7 billion. But gradually now it is decreasing. Now if you see per gene, even less than $1 per gene. Now you might be wondering, if you are not asking me, see, I say 500 to 5,000 USD per genome. So if you are listening me carefully, you should come up with a question, why, like for 1 genome, you are saying $500 or $5,000, right? So first of all, it is much lower than that now, but this 10 times higher thing is still applicable. How? If you want to sequence your genome on a medical purpose or something, your data should be much less redundant. But if you are doing it for research purpose, you need to get much accuracy, so you will be increase the redundancy of the data. And because of redundancy of you need more data, so you have to pay more. But on the other hand, these 3 techniques still, you have to take 2 to 3 years and you have to spend 3,000 to 5,000 USD. So why is that? In addition to talking about these techniques, we will be talking that, phenomenons, how it is coming in terms of cost. So we talk about X-ray. Now we talk about NMR also. What is the, how NMR is used to solve the protein structure. So when you use NMR, when the protein is hard to crystallize, as I told. For proteins that can be dissolved at high concentration, to study dynamics of the protein. As I told, the protein's motion is restricted when you go to the crystal. When you look at conformational equilibrium, folding and intermolecular interactions, you will be getting good information through NMR. Steps in protein structure determination through NMR: get pure protein, prepare the protein solution at very high concentration, because you need really high concentration for NMR experiments. Then do NMR spectroscopy, which is data collection. Then sequential resonance assignment. What is that? You go from, so how we collect data. First of all, what is needed for NMR? For NMR, you need nuclei with odd numbers. Like the best example is H1, right? So when you are going for protein structure, you are going for 3D structure, you need 3 elements to be behaving like that. One you already get, 1H, right? The other ones are generally N15 and C13. So you have to label it. That is one place where it is difficult to grow the protein, because you label it, bacteria don't see C13 or N15 much, because naturally it is N14 or C12. So there are a lot of situations where bacteria don't allow this protein to grow, or they don't grow by themselves. So first of all, when you do labeling, you spend a lot of time, like lot of money, but then you need even more money, because if a normal protein is giving you 20 mg protein from normal, 6 liter of culture, when you label it, the amount of protein drastically reduced. So those are challenges, but then you collect data. How you collect data? First you collect 2D data. You do 2 of the labeling ones and collect, and then you do 3D collection data. So you do all those experiments, then you collect the conformational constant and then calculate the structure. The advantage of NMR is, there are many, I talked about dynamics and all, but more importantly, in X-ray or cryo, you directly go to the 3D structure, right? In NMR, first you are calculating the secondary structures. So if you want to see the secondary structures, in X-ray and cryo you have to go to directly on the final structure, then you see the secondary structures. Where in NMR, it is stepwise. You get the secondary structures, then you reach to the assignment of the tertiary structures. Highly purified protein preparation, unlike crystallography, structure determination by NMR is carried out on aqueous sample. Usually the sample consists of between 300 to 600 microliter with a protein concentration in the range of 0.1 to 3 millimolar. The purified protein is usually dissolved in a buffer solution. So because it is the same solution where you are doing the other experiments in vitro, that is another advantage of NMR. So sample preparation, the structure determination by NMR typically requires a protein concentration of 0.5 to 3 millimolar, stable for several days at desired temperature, which is usually 20 to 40 degrees centigrade. Proteins and peptides should be dissolved in a suitable buffer 10 to 50 millimolar with 10% deuterium oxide. Ideally, we would aim for an acidic buffer pH 4 to 7, but dissolve the sample in a buffer that provide maximum stability and solubility in your sample. As the sample needs to be stable for days, the buffer content plays a critical role in protein peptide stability. So the enema structure of a protein is presented as a bundle of conformer. Why? In crystallography, you know the exact position of the atoms. So you get one perfect structure. Here you get the probability. So here in X-ray, you get one PDB file. In NMR, you get a set of PDB file, 20 PDB file in that way. And that's why you will see that NMR structures are coming as a, like ensemble of structures. Each conformer presents a good solution to the NMR resistance. First conformer usually is the base structure. So a lot of time you go to the PDB and you see the structure, you wonder which one to pick up. The first one is generally the high resolution structure, and that is to pick up. Typically a bundle of 20 conformers are deposited in the PDB. So I have highlighted some of the points about NMR. Some nuclei have non-zero spin quantum number, making them NMR active, but is the fundamental of NMR. Current passed through a coil induces a magnetic field, and a changing magnetic field in a coil induces a current. Placing nuclei with spin I equal to half into a magnetic field leads to net magnetization aligned along the magnetic field axis. So you have a external magnet and you have the small magnet inside the protein or any substance you are using. When you are putting the magnetization and resonance work, the small magnets would be aligned. When the B1 field is turned on, which is the external one, the net magnetization rotates down into the XY plane. Then the B1 field is turned off, the net magnetization relaxes back to the Z axis with the time constant T1. So that's the relaxation data you are collecting. Individual spin precises about the magnetic field axis. So this we are talking about 1D. When you have 2D, you have to do that 2 times. After magnetization is rotated into the XY plane by the B1 field produced from a pulse through the coil, it will process in the XY plane. The individual magnetization vector whirling around in the XY plane represent the changing magnetic field and will induce a current in the sample coil which has its axis along the x-axis. So what will happen when you are working with, you choose the elements, choose the like nuclei which you are targeting, right? Now you like look at the case, your case is going to be 1D. What is good for 1D? When you are looking for functional group identification in a small molecule, or when you are identifying a polymer which is generally not having a lot of different one or having a fold or something. You generally take the proton spectra. Now you might think how the proton would be separated. The proton would be separated by many things. First of all, with the chemical environment. So let's say if you have a hydrogen, the hydrogen is aligned to CH3 or CH2, it would be different first. Then how many peaks it will give also depends on, it is connected to how many proton it influences. So in that way our spectra get differentiated. So ultimately the nuclear spin produce the small magnetic field. Again the electron are spin I equal to half particle. They produce small magnetic field which oppose the external magnetic field. The surrounding electrons shield the nuclear spin from the larger external B0 field. This results in a reduction in the energy spacing of the two energy level and a lower Larmor frequency. This is called the chemical shift. So it is depending on the electronic environment, it is having an electron shield or it is withdrawing the electrons. Depending on that, you will see the effect balances out with the external magnetic field. In a frame of reference that rotates at the Larmor precision frequency, magnetization that is placed along the x-axis do not move, it simply relaxes back to the z-axis via the T1. So when you are doing it on the xy, it relaxes on the z. The effect is on the other one. So in that way it works. The nuclei with different chemical shift and Larmor frequencies will rotate around the z-axis at different speeds. T2 is the time constant for the magnetization vector to dephase in the xy plane. Then all the concept which we discussed leading to different 1D NMR experiments, combining them higher dimension experimental possible, which could be categorized into following categories. Homonuclear through bond correlation. Homonuclear means it's the same 1H with 1H, 13C with 13C in that way. Heteronuclear through bond and they are connected, that's why through bond. Heteronuclear through bond correlation. Homonuclear through space where it is not connected through bond, it is just connected through space, 3D. Homonuclear through space correlation methods, and a higher dimensional NMR experiment can be developed from the two-dimensional by incorporating additional indirect evolution time empowered with addition of NMR active nuclei. Like a 3D NMR experiment would be developed by performing experiment consisting of two 2D NMR experiment. The triple resonance experiment. The aim of the analysis of NMR spectra is to extract all available information about interatomic distances and torsion angles. For doing that, each resonance must be associated with a specific nucleus in the investigated molecule, which is called the assignment. So once you have the data, you put the data to map and find out where the atom is. Analysis of 2D data would be performed by using chemical shift, NOE. NOE is nuclear over-Hauser effect, which is how the H is affecting on H and all these things, coupling constants and amide proton exchange rates. Further in dimension, computer-aided structure calculation is to convert distance and torsion angle data into visible structure, mainly working in the principle of distance geometry and simulated annealing. So distance geometry is in this distance, it will be forming set of distance, it will be forming a secondary structure like alpha helix, beta sheets. Simulated annealing is when you have the data, how this like protein would be like going through heat. So heat is given to and it would be allowed to coming to fold again, and the energy would be calculated so that the among the different conformation the global minima would be calculated out. After solving the structure quality measures are performed using knowledge based method like comparing existing crystal structure. Measurement, RPF score, etc. using bond length and bond angle distribution, dihedral angle distribution, atomic packing, hydrogen bond geometries and other geometric factor. Finally, the structure would be deposited to protein data bank using 20 conformer, first one with the highest resolution. Coming to the third one, I know like because this is going long, what I didn't talk about here. I want to show you a movie and then we will discuss. So this is the grid, which is the part of cryo-EM, and then the grid is frozen here. This freezing is very important, and then the pictures are collected in the microscope. You know, it's an electron microscope at a frozen temperature, and that's why it's like a temp or a SEM, but at the temperature which will be helping the biomolecule to survive there. You get different conformations, and ultimately those different conformations, many different conformations, they would be simulated together to find out a three-dimensional structures. Some of the things which are very important and which make cryo-electron microscopy the success today they are getting. One of them is when you see there is a freezing, right? When there is a freezing, because there is, the sample is in aqueous solution, so water also freezes with it, right? When you are collecting those data, water contribute to the data strongly. So basically, more than protein, you collect the presence of water. Okay. So the challenge is you have to freeze it without allowing the water to be frozen. That is called vitrification. Vitrification is the key success of cryo-electron microscopy. So what they do, first of all, they do it in vacuum, they do in presence of liquid ethane, and they do it so rapidly that the ice crystal don't get time to form. Okay. Now when you are looking at the cryo-electron microscopy structures, who are the, remember I told when I was going there, it's a big range, but it's a microscopy, right? So smaller the protein, difficult it is to solve by cryo-electron microscopy. Whereas NMR is perfect for solving small proteins, cryo-electron microscopy actually working with bigger proteins. How big is? Experts used to say that if the protein is more than 1.5 kilodalton, then it is like easier to look at. But then if it is oligomer, then it would be easy. So 150 kilodalton protein and above that is easier for cryo-electron to solve it. So as I told, we talk about this money. So why so much time and money is required? I develop a cost comparison. So determining the three-dimensional structure of protein is essential for understanding their function and interaction. The primary techniques employed, we know now, X-ray, NMR and cryo-electron microscopy. Each method had distinct advantages and limitations, particularly concerning the instrument cost and consumable requirement. If we look at X-ray crystallography, the instrument cost, the initial investment of an X-ray crystallography setup is substantial. First of all, when I am talking about X-ray machine, there are X-ray machines for small molecule also. We are talking about X-ray machine to solve protein molecule. They are two different machines. Okay. In case of NMR, they are not different. That's what the beauty of NMR. So the cost is 2 to 10 crores for the instrument, depending on the capabilities and accessories, and additionally it facilities requires radiation safety measures and often necessitate dedicated space, which further increase the cost. Consumables is more or less normal here. From a target identification to purified protein, you need consumables to do that. NMR, the beauty of NMR is, NMR like crystallography is not using carbon, nitrogen, oxygen. Crystallography is only using carbon, nitrogen, oxygen, right? NMR, it could be any element in the periodic table. That's the beauty. I remember a story. The place I was doing PhD, University of Illinois, when I was there, it is having one of the best NMR facility, So there was a demonstration of a new instrument, and I still remember there was a NMR machine, and a lot of monkeys sit around and staring at the machine. That was the first slide Bruker people came up with. So we all gathered for this presentation, and one of our professor just quipped in a joking way that, so you think we are monkeys? And I still remember the answer this technical person he told, sir, with due respect, you are an expert, I know, but in terms of NMR as an instrument, we all are monkeys. Because one instrument with such a potential, applicable to everything with some setup, that is incredible. So the instrument cost, it depends on field strength. How the field strength comes? It comes in terms of megahertz. Okay. So if you think that you will work on a protein, then you need a minimum of 600 megahertz. But to be in safer side, you need a 700 megahertz machine. Okay. 700 to 1.2 gigahertz. That's what protein NMR used to be done. So 4 to 20 crores. Maintenance is like costly, but more importantly, as I was mentioned, when you are working with a protein to label it, the labels need restrictions, you need permission from DRDO, you have to buy all those, like C13, N15 isotopes. They are costly, and when you are applying them, the growth substantially reduced, then your cost increase, and all those things happens. The most interesting part is with cryo-electron. When I was a postdoc, not even a PhD, we used to see that cryo-electron is in very few places. India definitely had no cryo-electron. Suddenly what happened? Nobel Prize happened. And before Nobel Prize, NMR, like cryo-electron cost was average 250 to 300 crore. One machine. 250 to 300 crore. And suddenly Nobel Prize happened. Every country's politicians, they just told the scientists, we don't know, we need a cryo-electron microscopy machine. So suddenly from 5 centers, now worldwide there is nearly 1,000 machines. And the price reduced from 300 crores to, you will get a good machine in 10 to 12 crore, but very good machine at 25 crores. So I wrote 15 to 30 crore, and then sample preparation for cryo-electron microscopy. As I told, they involve vitrification using liquid ethane, requiring consumables such as the grids I was showing. So the grids are expensive, blotting paper, cryogens, and while the amount of protein required is generally less than that for NMR, the need for rapid freezing and maintaining samples at cryogenic temperature adds complexity and cost. So this is the second part of the presentation. The third part is about modeling. Thank you. So remember I told first X-ray structures came at 1959. You see in 2000 already there are good amount of protein structure present, right? That's why here you see a up. But from here the sequence started with the genome. But now you see how rapidly they are growing. So the first protein structure myoglobin was solved in 1958. The first protein structure database, the PDB, was established at 1971. The first organism to have its entire genome sequence was Haemophilus influenzae in 1995. The first protein sequence database was established in 2002. So I talked about the first protein structure database, which is PDB, right? But what is the first protein sequence database? This is UniProt. So PDB ID and UniProt ID. So what is the PDB ID? A ID which uniquely defines to a structure. What is a UniProt ID? A ID which uniquely defined to a protein sequence. If you know the PDB ID and UniProt ID, you will be perfectly hitting to the protein sequence or structure. On one sequence there might be multiple structures. Now when this data comes, being a lot, before that biology don't generate a lot of data. Biology experiments was long, they do a lot of work and come up with very small data. Whereas Human Genome Project changed everything and a lot of new data starts coming. Initially there was no database management. So what happened? Suppose I develop a database and I put it on the web. That's how I get the paper, right? And some of you, because there is no control, no management, some of you just copy from me, another guy, collate them, publish a better journal. You understand? So there was no control. People used to steal each other's data and all. And to solve that, the database management system came into. So a database theoretically refers to a set of related data and the way it helps the data to be organized. Access to this data is usually provided by a database management system. So once you incorporated DBMS into your system, nobody could steal your data. There is a process of, they could download it, they could see it, they could use it, but they cannot steal it. Okay. So DBMS consisting of an integrated set of computer software that allows users to interact with one or more databases. DBMS includes data definition, update, retrieval and administration. So how or what is the data is all about, how you could handle the data, how the data would be like regularly updated, because you will find a lot of databases which the people publish the data and then never look back. So the data is there, no updation happened. So if it comes under DBMS, it would be regularly updated and all those things, and there would be administration. So who are the databases? Primary DNA sequence database, DDBJ, EMBL, GenBank. Primary protein sequence database, GenPep, TREMBL. Curated database. What is a curated database? When you get a data and the raw data is deposited, that is a database, normal database. But when you work on that, suppose you get a gene sequence, you convert it to a protein sequence, you convert it to a protein structure, model, analysis. Those are curated database. And then we have the structured database. So now PDB contains 2 lakh plus structure. Be very specific. If today you check, there is 2,35,000 structures deposited. But we have billions of genes, hence we have billions of genes, protein sequence. So there is a huge gap between the gene sequence and the protein structure solved. But as we told, our goal is to reach the function, right? And the main gap is the structure. So how to get the structure? Our goal is to understand the function. So we need to do something to look at if there is some alternative methods are available other than experiments, because already we have seen that experiment takes a lot of time, experiments are expensive, right? So it is not being able to create or match the number which is coming through the genome sequencing. So we look at the possibility, and is there any possibility that we could do something theoretical process? And when we look at that, first the experiment which tells us that yes, you could do something theoretically is Anfinsen's experiment. So Christian Anfinsen, what he did a very simple experiment. He picked up a protein. This protein, in a presence of a particular molecule, convert that molecule from colorless to red. So if this is the molecule, my protein is there, it would be converted to red. Okay? If the protein is functional. That means if the protein is folded. Because when the time I am talking about, that time fold is confirmed by function. Okay? So he takes this protein out of the biological system, adds this thing, and he see that the molecule change from colorless to red color. That clearly means that the protein is active, hence the protein is folded. This time people have worked on protein folding, and they know that if you add denaturants, you will be destroying the fold. Okay? So what Anfinsen did, he add some denaturant so that the fold, protein fold would be destroyed. Now the protein is not folded. He again add the molecule don't turn red. So he told it's not folded and not functional. Now he start diluting the protein solution. So gradually the effect of denaturation goes away. Okay? And he then again take the protein, add it to that, and so that the color is coming. Very simple experiment. Protein adding to some molecule turns red. Denature the protein, cannot turn red. Again do something and turning red. Today you will say, what is the importance of this, right? This guy got Nobel Prize for this experiment. Why? Because this is the first time people realize that protein folding is not a biological phenomena. What that means? That means protein folding not happening inside the biological system. It could come out and it could see the physics and chemistry to fold. Means there is a amino acid which have to find a partner, right? The partner finding is not done by biology. It is done by chemistry and physics. That was Christian Hanfinson's experiment result. This experiment was supported by Levinthal. How? Levinthal told, so that time, I told you one colorless compound turned red, right? But that is the exception. Not all of the protein don't have that assay, right? And you don't have a CD machine to check the folding of the protein that time, right? So other scientists start talking that probably the experiment Hanfinson have done is an accidental one. It might not be applicable to all the protein. To resist that, to argue again in support of Christian Hanfinson, Levinthal, his friend, a mathematician, come up. He told, let's imagine that there are 200 amino acids. He don't know like us, right? He don't know 300 was the number that time. So he told, let's say one average protein have 200 amino acid. Let's take one amino acid. What would be the minimum movement? It would be going this side, right? And this side. So how many conformations? 3, right? So if there is 200 protein, 200 amino acids, the total number of conformations are 3 to the power 200, which is 26 into 10 to the power 95. That many conformations have to be taken. So let's imagine one conformation take 10 to the power minus 10 second. Still it take 3.2 into 10 to the power 30 years. So if, as Hanfinson told, the folding is driven, if it is not driven, if it is doing with everyone, then it takes that much time. But we know that protein folding does not take that much time. Hence, Christian Hanfinson is right. This guy also got Nobel Prize. So 2 Nobel Prize, and you all understand, right? Now I have to say something. When I studied, like my teachers also told about these stories, I believed. But when as a teacher I came to, I have few problems and I will discuss with you so that there is nothing bad with that. Both of these guys did great work. But I have something to tell you what I realize as a teacher. Okay. So first going back. And Hanfinson told protein folding is a physico-chemical phenomena, like it does not need any biology, which might be correct in a in vitro solution. Are you guys understanding what I am saying? In a beaker, in a big volume, there is time to relax. There is time to make mistakes. But think about an E. coli, total 2 micrometer cube volume and 5,000 protein, ribosome, so many organelles. Is there place to take those or make those mistakes? No. You understand what I'm saying? So, in a in vitro condition, Christian Anfinsen theory is very, very correct. But protein folding happens in two ways. Protein folding happens in your laboratory without any biological influence. Protein folding happens inside your body. Both are giving same results, but they are not going through same path. Are you getting me? In the biological system, because there is molecular crowding, you cannot get the opportunity to look at the interactions. If you have to look at, you take much more space, right? And that's why when you read biology, you will see something, a group of protein called chaperones. Chaperone proteins are there to help you fold. If Christian Anfinsen was totally correct, you don't need any chaperone protein to be there present in the biological system. So protein folding resulting same result, but going through two different mechanisms, inside the body, outside the body. Is it clear? Clear everyone? Coming to Levinthal. आप सब बैठे हुए हो? You are all sitting here. When, what is your name? You. Paramita. When Paramita is moving, what is your name? Aditi. Is it influence Aditi's movement? No. Right? So, when Levinthal told, when one move, other will not see sitting there, they will, both Paramita and Aditi could move together. So the number calculated is not exactly correct. Though it's true that the mathematics he wanted to do is correct. That without any directed phenomena, protein folding is not possible. But the number he have created, the basic of the mathematics is wrong. You understand? That's the thing. The third logic, which, so these two are giving us a base that it is possible probably that protein structure could be done through theoretical methods, right? The third one is even more beautiful. Remember I told you get the protein crystal, you diffract it, you get intensity. Intensity is I, need to be added with the theta, the phase, right? Phase is needed. Now, how you determine phase? From some reference, right? Think about, in the protein crystal, who are there? Carbon, nitrogen, oxygen and hydrogen. Hydrogen don't diffract. Carbon, nitrogen, oxygen are mass wise, electron wise, charge wise mostly same. You cannot differentiate in between them, right? So that is called the phase problem. How you solve this phase problem? Generally crystallographers put a heavy metal. What happens then? Sometimes the crystal spoiled. But after doing a lot of them, one crystal at least stay there, in reference to that they get the phase. Okay? Good process. This is called the metal based phase determination. Possible theoretically? Problem nahi? No problem, right? But when you are working with metals, most of them are heavy metals because you want to differentiate between carbon, nitrogen, oxygen, so you take lower periodic table metals, right, which are heavy. Heavy metals are radioactive. What is the result? The first generation crystallographers, 99% died in cancer because they handled those heavy metal in hand. It's a huge terror. People didn't wanted to do crystallography. It was that type of situation. And then came this guy. Why I say this guy when it is written something process? Because it is his signature. Molecular replacement is the signature of M.R. Michael Rossmann, one of the most successful crystallographer of our history. But the beautiful part of that, he was a very good theoretician, very good programmer also. He told, I could come up with a process of phase determination. Say, how you could do that? He said, let's say this is the salt structure of one protein. Already someone solved it. Okay. Your protein is 90% identical to that. Are you, by the way, skeptic ki why 90% and not, lesser? So let me tell you, that time even 99% was required. Why? Because you crystallize a protein, you solve it, right? What about the function? You have to do mutation. So if you do one mutation in a 300 amino acid protein, what is the percentage? 0.33. So this is 99.66% identical. So 90 is pretty good. Now you understand? So he developed a process where he told, this is the salt structure, you align it. So let's say this is the unsolved structure. What I have to do to align here? First I have to bring it here, right? Any geometry you coming in your mind? Correct. Translation. So for 3D, 3 translation. So yeah, it is here. Now what? Rotation, right? So you could do 3-fold translation and 3-fold rotation to align it and take the phase of this guy. And this changed the world. He solved it by doing this process of molecular replacement. Someone in the audience is skeptical. Sir, just finished crystallography, that time didn't talk about that. Why you are now talking about that? Because there are 2 functions I talked about. What are the 2 functions? One is translation, another is there any experiment? No, both of them are theoretical method. Now that gives theoreticians a thought. Oh, if an experiment could be solved by a theoretical method, why not a theory could be solved by a theoretical method? So these having a biggest influence on going to the theoretical prediction of protein structure. With that, people have gone collected a lot of information about the side chains, how the side chains are rotate and all of these. They develop rotamer libraries and let's say you take 2 amino acid, Ella and Arch. So you get 3 combinations, Ella, Arch, Ella, Arch. When you see, you check the Ramachandran plot, you see that they are behaving very differently. So one neighbor amino acid have so much difference, you could calculate using theoretical method, then theory have a chance. And more importantly, do you have 2 pens? Take 2 pens, show me. 2 pens you are having. Hold by 2 hands. No, both of them 2 hands. Acha, मैं demonstrate कर देता हूँ. Come. So 1 pen, rotate. Is it rotating? Second pen, try to rotate. It is rotating. That means now if you look at DNA, RNA, carbohydrates, all are connected by single bonds in their critical polymeric junctures. Think about protein. Which bond? Peptide, CONH2. It's a double bond. So it restricts the movements. And when there is restriction, there is chance of theory. Because if you don't listen to your parents, then they have no control over you. Right? So here, proteins start listening. So theory is possible. Right? So there is a possibility of computational modeling of protein. So a predicted model simply illustrates our assumptions. When you have X-ray crystallography, no assumption, this is nature telling us what is there. But when you do a modeling, it is a computational technique and it's an assumption. There are three major methods. I'll pick them quickly because in tutorial I will talk about. One is homology modeling, second is fold recognition, and third is ab initio. Homology we have talked about, but if you think and when we go there, remember that advice, accurate modeling always does not depend only on greater sequence identity, it depends on the similarity between folds and functions of the template and the target protein. So what is homology modeling? You have a sequence, you want its structure, you search for and you get a homologous sequence who have structure. You use that structure as a template and do the modeling and get your structure back. This is the process of homology modeling in brief. In details, you first need to recognize identification of any well characterized protein similar to the protein you want to make structure by using blast search. Position by position target template equivalence sequence alignment. Then when you have alignment, why you need to align? You will see that sometime you get 45%. 45% is a good one, but is it whole through the protein or it is a domain? You will only understand when you align them one by one. Okay? And once you do the alignment, you go for modeling, detailed 3D structure of protein using the inbuilt algorithm. What that means? Every program have their own inbuilt mechanism. Okay? And then once you get the model, you do the model analysis. The model protein can be verified by different softwares. We will do that. So for recognition of template, blast, SciBlast and all, identify the PDB code of the template. That PDB ID, remember I told you need a PDB ID. What is a PDB ID? PDB ID is a combination of letter and number, starts with number. So it starts with 1, 2, 3, 4, anything. What is the significance? If you see the number, you know that the structure solved in what time? Like if it is 1, it is solved in 1960s, 70s, 80s. You know what's the number now? 9. Which means you guys are in the transaction of the time when this 4-letter code of PDB would be changed to 6-letter code. It will be happening within few months. Actually, it is planned already. People are working on these and all. What is UniProt ID? It is again a number and letter combination, gives 6 letter and number. But if PDB ID is going finished, think about what would be happening to sequence, because every day genome sequencing project is giving you thousands of new sequences, right? So they started with 6, now they have 10. So UniProt ID is a combination of letter and number, like 6 letter and number or 10 letter and number. So here you have to have the PDB code, and then you have to do statistical significance of the match, which is by Z-score, E-value. So what are they? E-value, the number of hits one can expect to see just by chance when searching a database of a particular size. You guys came from different places. Now you get friendship. Now you stay in very different place. If then the chance of you guys meeting in one of your favorite shop is very rare. So if you meet, then there is some story behind, right? So that's what E-value is all about. Z-score, this score expressed as a distance from the mean calculated in standard deviation. If you see, Z is the standard and how much you are deviating. You have done it in statistics, right? Now in blast search, we will do blast search. What you will first see? You will first see the query coverage. You know, we organize some projects and all, a lot of people come and they make a common mistake sometime. They are making modeling. They say, sir, I got 80% identity. Then it should be a good protein, but then we ultimately find that they actually didn't see the query coverage. So it is query coverage 50% and among them 80%. So basically it is 40%. So your identity is query coverage into identity. You understand? So it's very important to get the query coverage. We'll talk about them. Alignment, there are softwares like Clustal W, T-Coffee, Clustal X2 for modeling, for commercial programs which we have to buy auxiliaries and typos, but free softwares, Modeler by Andres Sully, Jackal, Barry Honing, Scroll, Ronald Dornbach, and Swiss Model. Today we will perform through Swiss Model. Then verification of model quality. How you verify? One, geometric, one, Ramachandran plot, and second, the energy. The application of homology modeling. Why you are doing homology modeling? Structural knowledge of protein gives some understanding of function. If you get the structure, you could have guessed the function. That's what we are talking in the whole talk. It helps to understand the mechanism of action, like how a structural protein binds or how an enzyme works. Predicted structure can be used in structure-based drug designing. It could help us understand the effects of mutation on structure and function. And it could be used in comparative structural study between the related proteins. So what are the limitations of homology modeling? Small molecules, waters and ions used to be retained from the template structure. It used to be. Now gradually the programs are improving, and you will see that especially metal ions, which are very important. Generally you have a metalloprotein, you model it, you will see the metal is also coming. So the modeling algorithms are improving. Experimental data is not incorporated, so you don't like incorporate those. Especially the mutagenesis, which is very useful, you know, by changing the amino acids and all, what will happen. The presence of the ligand is also likely to alter, like when the ligand is coming, especially when protein interaction is coming, there is changing of conformation. So that is not possible to trap in modeling method. electron density of the selected model. So remember I talked about when we do diffraction we get a electron density, right? So when you are choosing a model, you are choosing based on homology with the sequence. So you are not looking at the electron density, right? So sometimes you come up with bad structures. Okay. So two zones of homology modeling. The first one where higher identity is there is a safe zone, but lower one it is more difficult. And if you see 40 to 100 it's easy to model, 25 to 40 it is difficult, and below 25 it is not possible. So there are many differences, like you see these two, these two proteins, phosphoribosyl transferase and viral coat proteins, they have around 42% identity. So they could be modeled from one from another, but it is not possible. Probably because they are covering small area. Then it also introduces biasness. What is the biasness? You see, this is the template structure. In the template structure, you could see the valine, right? Now, when you come here in the new structure, you get glutamine. See, the glutamine is getting the same side chain rotamer conformation like valine because it is biased. So what to do? So if you do modeling, you cannot correct it. You have to do modeling followed by energy minimization. What is energy minimization? So we understand the basic concept of physics. If you lower the energy, at the lowest energy the molecule represent the natural state. So you keep the energy lower and you will get the conformation, like you see it is changing the rotamer conformation. Some molecules like histone H5 and transcription factor, both of them are DNA binding protein. But if you see their identity is only 7%, but they are very similar in structure. So they are exceptions. Then there are multiple conformations. One protein could be stayed in two conformations, like a channel protein could be stayed in open chain and closed chain. Which one you have to choose? How you choose it? If you don't know what function you are going to explain, you cannot do it. So a theoretician should have a good understanding of what the experimental process going on. If you don't manage to get both of them, you have to do theory, but you don't understand at all what the protein is doing, what is its experiment, what are the experiment process, you cannot come up with a good model. And then loops are always big issues. Why? Because even experiment, loops cannot be collected with good data. Why? They are very flexible. So let's say X-ray is diffracting. The loop, one residue is here now, going there, you know, it's moving. Because of that movement, the experimental data itself is not good. If experimental data is not good, how the theoretical data based on this experiment could be better? So this is a very big problem with the loop modelings. Beside this, we have two techniques. One is existing fold and another is new fold. So I'll brief the process. Because existing fold, that is called threading. Why threading comes as a process? Because when PDB started in 1971, every day new structure is coming and people could see new fold. New structure, new fold. After around 10,000, 20,000 structure came, new structures are coming, but no new fold. Why? The initial story I told. Nature did not take the sequence space. Nature make its very small conformation space. Nature have to do so much work. Right? So nature didn't create individual fold for individual protein. What nature have done? They have make some of the modules and now adding A and B to one protein, B and C to other, A and C to other. In that way, same fold is distributed to different proteins. So if that is true, then if we create a database, can this database could be helping us to solve structure? Yes. That's what is GenThreader all about. Ab initio is another very interesting one. Here you don't have any information from the experiment. What you do calculation. Okay. So you have a equation and a problem. You optimize it. You don't use the experimental information of others. What you do using quantum mechanics. Now, people who do chemistry, they could understand this. 10-20 years earlier, people have to do structure of all the small molecules. Now most of them are DFT calculated, right? So similarly, you could have done the calculation in the level of electron. The problem is proteins are bigger. So you cannot calculate the entire protein, but you calculate a small part. A small part of protein is called peptide. So before, peptide was not a very important thing. But nowadays, people are looking at natural foods, food-based therapeutics and all. So peptides are coming very important. And peptide structures, peptide folds, peptide behaviors are done by this ab initio method. So these are the three different methods, and then all the methods are kind of coming together. I finish it. After lunch, I will explain these mixing methods and do the tutorial. Thank you.
Thank you, Professor Hajra, for his detailed and insightful lecture on protein crystallography. Now we will go for lunch and we will come back again, and after that we will complete this session, and question answer session also be open for you. Thank you.